Hi class. Uh, Please join here. I think voice is better than before. I'm very much sure. So we again started. So 10 minutes passed. Sorry for the 10 minutes passed. This live streaming is the first time that I'm doing from my house. If I think doing it from the home is it's still buffering. Hmm. It's because of the students. Earlier, if we have few students, then it was fine. But having a uh, traffic coming there, it's letting us be like this. Let's start. It will be fine, I guess, with the time. I hope so. Internet problem. So I have switched all others, other platform who were using internet my house, closed everything. So earlier the problem was my voice, but now my voice is fine. The buffering is happening. You cannot see anything. Yes, it was when we started. Okay, okay. That's bad. Very bad. Let me just check. I cannot change the system now. I hope the internet connections gets better. I start giving my present like who I am, something about your trainer. Buffering. Yes, it is buffering. Okay, voice is coming. That's good. That's one thing is at least good that we have to be see half glass empty to be that it's still half glass is full. It is not clear properly. Okay, let me talk to my head about this problem. What they suggest. Some other platform like Zoom. Zoom is only for 100 people. WebEx I never heard. Uh, Microsoft Teams only 100 people. We have checked everything. Let me just have a small chat with my boss. You can't hear anything. Sir, there is a problem of buffering. I told you already before. So as the traffic is coming, it's becoming very slow and the screen is not coming fine. Buffering. Okay. So I will start with little bit of my introduction. Um, who I am, where I'm from, why I'm here today. These are the basic questions we have: who we are, why we are here for, what is the reason being uh, who we are. So I am here because I love to teach. That's my passion, and I love to learn also. Um, I always keep learning from all different perspectives, whether it's a sports or mental problems or any uh, or any other parameters that are related in our life for being a better person. So always keep learning in my life. So this is something about me from my education perspective. So I did my bachelor's in biotechnology with 67.4%. So I'm an average student. I was from Shahid Singh College in Chandigarh in 2009. Then I got a chance to be a lecturer for 5 months at the Ben Singh College in Gurdaspur for 5 months, where I was teaching bioreactor designs, environmental and bioprocess engineering. And then I went for my master's to Germany at the Justus Liebig University, where I had got chance to do a project with Max Planck Institute of Heart and Lung Research, where I worked with the metastasis, how cancer cells exacerbate our tiny normal cells to go for, and where I got second highest grade in my master's. Then I've switched to my PhD in cardiovascular physiology, in molecular medicine field, where my project was related to with the redox regulation of two proteins, PP1 and ER stress regulation of connective tissue growth factors in cardiomyocytes. So I was basically dealing with the heart failure topic there. So that was done in 2017. Then I came back to India. Then I joined as assistant professor at Mata Gujri College for like almost 1 year, and then since then I to keep my because what I saw that research is also good, R&D part was also good, that having a foreign experience, but within myself there was always a race or some rage that I want to teach students. I have knowledge, I have experience to my maximum level of education. Now it's time to provide this to student platform. So I start teaching on freelance basis, class 11 12 for the NEET preparations. Also joined recently because of COVID time, we all are becoming now online professors. So I got chance in July giving a biochemistry workshop of on biochemistry, and now I'm teaching with you guys today with MBBT, and most likely we will go over other workshops soon. Also, I'm also associate subject matter experts at Evelyn Learning System, which I recently joined. So this was my publication in Science Reports. This was done in 2018, where I worked what I did my PhD was successfully published. Um, Beyond that, I'm also fluent with German. Hallo, wie geht es dir? Wie geht es Ihnen? Und was ist heute hast du gehabt? Und welche beste ist deine Hobby? So ich verstehe, ich kann sprechen ein bisschen Deutsch auch, while staying in Germany. So if somebody speak German, please let me know. Yeah, it is fine now. Yeah, people are saying it's fine, everything is fine. Just uh, voice is fine, everything is fine. It's now clear. Just calm down, guys. Uh, just listen and don't be exaggerate things. Just yeah. So these are the something about my by being trainer here. So what we will be doing, we will be doing today some basic principles of MBBT. Why we are studying molecular and biochemistry techniques? What is the reason behind it? Then the second day we go for cell culture and microscopy. We will do various presentations, some videos, some protocols. Yeah, so playing some game also with microscopy. There's also games here in our things. And then so basics, your MBT techniques will be done in this case. Then recombinant technology techniques will be done on the day fourth. Then we come after dealing with the DNA and RNA. Now it's time for the proteins. So on the fifth day, we deal with the protein quantification, separation, and purification. Then we deal with the electrophoresis part of like gel electrophoresis. We will also have some virtual labs to do so. Of the previous workshop in the biochemistry, we did 3 workshops, so might be continuing the same here sometimes, and with some more protocols also this time. Earlier, I didn't produce any protocols to you. Then yeah, some blotting techniques, which is quite important when you enter to the lab. So there will be a lot of things from northern blotting, southern blotting, western blotting, yeah, all north, south, east, west. So you will be doing these techniques there in the lab. Then FACS analysis, your flow cytometry techniques for to analyze cells will be also be taken care for. Then something about I want to touch a little bit from my PhD thesis, bioinformatics, how it could be important for you while staying at home, how you could be a computational biologist doing so many things on your computer. Then on the 10th day, some applications and recombinant technologies and or applications of your molecular biology technologies. So some applications of what your techniques are doing. So on the last day, we will learn about their applications. So in 10 days, it's quite hard to add everything. There could be a lot of other techniques I want to add, but I think I will add also there's something I want to teach you about NGS. Next generation sequencing, because in this COVID time it's very much important. Yeah, it's very much likely that everyone is working with the NGS to find out the genome of our, yeah, COVID-19, which had created such a pandemic in everyone's life here. So to do so, next is your questions, your doubts were coming about uh, Danke, gut und Sie, guten Abend. Yeah, learn German. Yeah, if you have any questions later on to seek for your study purpose, I'm always there. You can ask. And also, please don't forget, if you like these workshops, go for another workshops where you will be dealing with principles of plant biotechnology and genetics techniques, where like before I go further, let me explain about projects here. So it's around 303, 304 viewers at the moment, and I want yeah, you are doing all everything in collaboration with IIT Kharagpur. So in your projects, what I have decided is each day when you are done with your let's say let's say you are done with here day one, you're done with the basic principle of MBBT. So I ask everyone to make a two-page. I will also write it later after a workshop. A two-page assignment about the summary. What have you learned? Only two-page, not more than two-page. That's the task. Only two-page summary. What have you learned? Whether you want to use an image in your own words, taken care of plagiarism. I would not like any copy paste thingy, because we can caught this very instantly that it has been copied by Wikipedia or something. Do it on your own words, and by doing so there will be 10 days, so it will be our 20 pages assignment. So in your project you have to write from the first day 2 page each day, what you have learned, what are the principles that you have learned, and your inner task that you feel the most that you can save for yourself in the future. Yeah, that's the main task of this project, is that whatever you learn from us, that you can write in your own words for yourself, and whenever you're going for an interview or anywhere, you can it's on your tips. You just open this 20-page things, or you, of course, you can go through our workshop also again. But this will be your own efforts and your own way to learn things in a good way. So remember that every day write 2 pages what you have learned in a summary with the images and methods, principles. You feel like the best thing that you can remember out of it as a juice out of it, yeah, like Musambi's juice. So whatever the nectar you can get out of it, just add into that in your project. So that will be your thingy. Guten Tag, yeah. So Uh, if somebody is also being part of internship for them, I will explain after third day what they can do, because there will be some case studies with reference to the research papers. So just write me over your on the Viber, or what was it? Telegram. I always forget this name. Telegram. So write me on your Telegram and we will talk about this. So it's 26 minutes. I have wasted 10 minutes before yours. And I would like to start our first session. So if we are like 15 minutes late, so I will take the class 15 minutes more then. So just take care of that. So I will not leave you. I wanna give you all the things that I have here today. So one clap, shoot out to the uh, okay. Anyhow, so before we start today about our workshop about molecular biology, it's good to understand what is cell is. Yeah, because this is the basic foundation of our body. If our cells are working, our tissues are working. If our tissues are working, our organs are working. If organ is working, our organ system, total whole system is working. Then our body is working with all the organs together. Then one system is made. Then our bodies is likely to be in a good mood. Then, so how we can say that cell works like a factory? Is it? Can we say that? Um, because in a factory, let's say in a shoe shop where Bata shoes are being made, Bata shoes, let's say, and there are workers, there's a boss, there's a gatekeeper, there is a some free time area, there's a where's a production of Bata shoes are happening, where there's a labor who are working in them. There are people who are dealing with the transport system within the Bata factory. So all these things are also actually happening the same way in the cell also. So that's how cell. So that's the yeah, the synonyms, or we can say, what is the, I forgot the word in English, like the mirror of each other. Uh, so this is normal structure of cell. You might all be aware from your class ninth or 10th, where you are taught about cell, which is a unit of life, and you were told about lysosomes, nucleus, centrioles, rough endoplasmic reticulum, nucleolus, nucleus. All these parts are being taught since past. But before we start, I want to start with your basic, because some of you might be of different background, some of you might be really expert in this. So for me, if I'm taking this workshop, I want to take you as granted you are my students, and you might be not aware of. So I have to uh, start with something basic now. Before, if we start something, if our basics are not clear, it will be not a strong foundation. So something, if somebody knows, there will be a refresh of their memory. For others, yeah, it's good to revise things. Okay. So, how a cell can be like factory? What do you say? How it can be like factory? So I have told a bit. What do you say? Oh, if you students are into the assignments, just calm down. It will be online. Uh, so how cell could be act like a factory? Tell me about this, students. Yeah, how it could be? Yeah, analogy. Yeah, that's the word. So how is it? Any answers? No answers. Bad. So in a factory, if we have many different tasks being completed in a same building, so many tasks that are being done in a factory, they're done in the same building. So let's say supporting and sheltering the workers, workers build goods, maintenance crews clean up messes, shipment department is there shipping things from here to there, and so on. So there are a lot of work is happening in the factory, and cell in the same way is doing different department work in the same in a specialized task way and performing in a very competent and very stringent way as it is. The cell wall is like a security gate which does not let enter anything. So security gate protects the factory and helps keep it safe. Yeah, a cell wall does the same thing for the plant cell. So it does not let enter things and in and out like that. And cell membrane afterwards is like the front doors. After you enter the main door, then there will be doors of your house which will enter and leave the people of the building in the factory. In the same way, in the cell membrane for the plant and animal cells, we have these doors there. So nucleus is kind of a CEO, is a big boss of the factory, which acts in the inside of the cells, and it's the same do for the plant and animal cells. So the big boss is there. So this is how it is. Then further, cytoplasm, which is an empty area of the cell, is the same way as the factory floor of the factory where workers, machines, equipments are working on, and cytoplasm holds all the organelles and the plant cells and animal cells in it. And endoplasmic reticulum is the assembly line where everything is being moved on the conveyor belt. So different parts. So like as I said, example of Bata shoes. So this NDER, what is it doing? It's helping to move your stuff from here to there. So like this. And ribosomes, they are the assembly line. So they are people, they are building factory. So your protein is being produced in this case. So ribosome assembly proteins are happening in this case. Cool. Vacuoles are the places where you have your free space where everything whatever is not needed could go. So they are like break rooms where you have storage of food items and place to get rid of trash. So they hold waste and stores water in plants and animal cells. So which one has one large vacuole? I think it is. Which cell has a one large vacuole, students? Yeah, yeah. Which cell has the largest vacuole? Plant cells. Good, you are all are good in it. That's good. That's good to see. So next part, lysosomes. They are like janitors. You know janitors, which works in the factory for the maintenance of the things. The one person who can fix everything for you, like break down, clean up any repair items. So lysosomes works the same way in the cells. So this is quite nice analogy. I tried my best to bring it for you. So mitochondria and chloroplast, they are the like ATP, the energy energy production of your cells. So power station, the power station which supplies energies in the factory. So mitochondria in animal cells and chloroplast in the plant cells do this for a cell. So here you can see an example of your ribosome being on the conveyor belt, and then out of that the mitochondria from the heat source is coming, your equipments are being produced, and inside this is a factory, is the nucleus and chromosomes are being residing there. Then on the other side the Golgi apparatus is being there to remove your waste out of your things. Then there's a protein production is happening out of the ribosomes, and there's a ER to movement of the things, and this is a restricted area, this is your cell membrane outside, and this is in and outlet of the cell. So this is how a factory could work um, as a cell. Yeah. So this, in this case, you can draw one assignment. This is very yeah. In the first case of today, you can think of something that makes. You can design a factory that makes something and assign each organelles like on a worksheet. Yeah. So these pictures could must present their job, what they are doing. So like in a ketchup factory, the proteins should be the bottles of ketchup. So if we can show that these bottles are being produced as a proteins at the end, and you can see from this example, previous example, how a factory design is made, and same way you can draw one picture. So that will be for this part for today, and then I would like to take you to the other part of it, that is cells. So here I want to show you how much size of a cell is actually. How much size is the of our size of our cell looks like. So this is written is your times regular 12 point. So this is your written normal times regular 12 points. So your coffee bean is size of 12 by 8 millimeters. Okay. So you just keep seeing things now. So your grain of rice is 8 by 2.5 millimeters. Okay. Your sesame seed is 3 by 2 millimeters. Then your grain of salt is 0.5 millimeter. Okay. Then we go deeper. Amoeba proteus is around 500 micrometer. Then comes the paramecium, which is 210 by 60 micrometer. Then comes your skin cell, oh sorry, human egg, which is 130 micrometer. Then the skin cell, which is 30 micrometer. Your photoreceptors, that is in your eyes, 2.5 to 100 micrometer. Then your sperm cells, around 60 to 5 micrometer. Then your red blood cells, around 8 micrometer. Now comes the chromosome, around 7 micrometer. Baker yeast, 3 by 4 micrometer. So just keep seeing. So we started with the times New Roman 12 point, bullet point. Yeah. So E. coli 3 by 0.6 micrometer. Lysosomes 1 micrometer. Mitochondria 4 by 0.8 micrometer. Now comes the viruses, so even smaller. Measles virus 220 nanometers, they are in nanometers. Influenza virus 130 nanometer, HIV 130 nanometer. Hepatitis virus 45 nanometer. Rhinovirus 30 nanometer. Yeah. So comes now more small parts. Ribosome 30 nanometer. Antibody 112 nanometer. So your transfer RNA 7 nanometer. Your hemoglobin 6.5. So let's go more. So I hope you know all these names. If not, we will go through it now again. Your phospholipids 0.9 to 3.4, 0.3.4 nanometer. Glucose 900 picometer. Your adenine, your methionine, you see they are in picometer now. Your water molecule 275, carbon atom 340 picometer. So we started with the meter and ends at the picometer. Can you see from the carbon atom to all viruses came into nanometers. Then comes your bacterias in micrometers. Your cells comes in micrometers. Then millimeters. That's the story. That's just I want to explain how naked we as a human being, how our smallest micro particles we have discovered from meter to picometers. So far clear? Science is exciting. Um, Science is exciting. So you learn a lot from your when you start to learn about it. It's so much, so many things you can explore. You just need the right platform, right place, and you can start with it. Yeah. So let's continue the next part. So, bache, some students are asking me where you have to send your assignments. So you can send me on my email ID. I will share it later after the workshop is done on the Telegram, and you can send your assignments after 10 days is over, not now. So not every day you are sending me your 2 days, your 1 day written 2 pages things. It has to be together, and I will happy that you should not write it with your hands, something written, and it has to be written via computer, or you can type it down, not written one, and please do it so. Okay. Who is this Sonura? Please don't share these smileys or so. It seems not so professional. So I will let's not stop and continue for the second part of our today's workshop about the basics of molecular biology. So, as we have discussed cell, what is cell is made up of, we have studied the analogy, and we have studied the how cell size could matter, how minimum to the largest cell it could be. So here today in this part we will dealing with the basics of molecular biology. Before we start with started to speak about molecular biology, so this figure is just showing you what is the difference between eukaryotics and prokaryotics. So students, this slide that I have made for you, this one is basically to have you all the basics clear from your biochemistry, microbiology, molecular biology. Your basics should be clear before we start further. So it's a rebrushing your memory. That's my main motive to teach you today. That's the main idea of today's workshop. Okay, so organisms. So the difference between eukaryotics and prokaryotics. In the first one, the eukaryotics includes your plant, animals, and fungi, whereas in the prokaryotic, it's bacteria and cyanobacteria. On the other hand, Yes, animals have no cell wall, whereas plants do have. Prokaryotes do also have cell walls. Chromosomal segregation is done by mitotic spindle case in the eukaryotes, whereas the chromosome segregation is done by the cell membranes in the prokaryotes. So meiosis is happening in eukaryotes, whereas in prokaryotes it's not happening. Ribosome size is 80s in the eukaryotes, whereas in prokaryotes it's around 70s. Now something about cell organelles. So all these nuclear membrane, ER, Golgi apparatus, mitochondria, chloroplast, they are all present in the eukaryotes, whereas they are absent in the case of prokaryotes. What is the definition? How we can define molecular biology? It's actually a study of underpinning of the process of replication, transcription, and translation of the genetic material. So all the process, that is your replication, your transcription, your translation, that is known as central dogma. From DNA, transcription happens to make your mRNA. From mRNA, your protein is made with the help via translation. And this whole thing, to study all this process of production of RNA and protein via technique of transcription and translation respectively, is known as your molecular biology techniques. Um, so it could be interconnected with others field of science also. So let's say, like in this diagram I have shown you the cells, plants, microorganisms, molecules, population, animals. They're all co-interrelated with these studies, with cell cycles, extremophiles, enzymes, structural molecular biology, genomics, mathematical biology, entomology, with the insect studies, membrane traffic signaling with the molecular biology inside it. Yeah, plant development and defense, how the gibberellins and all these defense systems are working in plants. So all are interconnected, and to study all these parts is known as your molecular biology. So the field overlaps, so this field of molecular biology overlaps the biology and chemistry, particularly with the genetics and biochemistry. So chiefly, it is told that it's understanding the interaction between the various system of cells, including the DNA, RNA, and protein biosynthesis, as well learning how these interactions are regulated, as we have discussed it already before. And it's also dealing with the yeah, quantitative. Much of the work of the molecular biology is quantitative, which has been interface with the molecular biology and the computer science in the bioinformatics side. So it's sometimes quite hard, very hard to study such big genomics data of our human body. If somebody is working with a gene therapy or cloning or site-directed mutagenesis or to with the Cas9 recently came now CRISPR Cas9 thingy, and now at the moment people are working with the COVID-9 virus. So it's very hard to genome everything by ourselves to see the genomic data. So for that, this computational biology, this bioinformatics plays a big role, which brings our huge data into compact form, and with the help of softwares, we can manipulate with the genes and look for the effect directly, what happened to the proteins. So this is quite yeah, up to date and quite demand in the future for the bioinformaticians. So how the molecular biologist field has started? So it's in 50s and 60s. So there was characterization was happening, how to isolate and manipulate the various in the various components of the cells, including DNA, RNA, and also protein, and to check their structural and enzymatic properties in the cells itself. So in the 50s and 60s, this field of molecular biology has been started. So this is the main components that are involved in the molecular biology, that is DNA, RNA, and protein. So your double helix structure, you all know this double helix. This is your RNA. It's sometimes double helix, you can see, sometimes single stranded, and then there's this loop of there, and then again single stranded. So this is the RNA typical structure, and this is protein quaternary structure, having beta sheets, alpha helix structure. Uh, yeah, all being coiled together. So from your DNA, RNA is made. From RNA, your protein is been made. So gene, what is gene? Gene is a unit of heredity. From here, your Mendel, the father of genetics, Mendel just produced this unit, this definition, this term at that time with the gene first time from while working with the pods, which you have studied in your 10th class, I guess. Uh, it's quite good like how our CBSE system or somebody other boards was learning we are told and taught about these major biology parts since our school level and which continues to your even PhD level. So I'm really helpful to our Indian system of teaching science that whatever I have been given information in my BTech in India since in my school level also, it has really helped me till now in my life. The way the syllabus has been made, so all information has been taught in very good manner. I have to admit that our education system is quite strong as compared to foreign countries. So let's to continue. DNA segments that carries genetic informations are called as genes. So here you can see this is your nucleus. Yeah, in the nucleus you have your chromosomes, 23 pairs of chromosomes. Um and then from the chromosomes they are coiled around these are coil your DNA is coiled around histones. These are histones molecules. There are four like histone A, B, C, D, four types. H1, I forgot the names. So they're in the four chambers actually. So then they are coiled together, and you can see uncoiled version of that. So this is how from your cell chromosome, then on histones, your DNA has been coiled, then it's like uncoiled version. This is how your DNA is coiled within the nucleus of your various 23 base pair of chromosomes. Gene holds the information to build the maintain organisms and pass genetic traits to offspring. So like this. So if we are doing the karyotyping, so you see some dark regions and some light regions. So in the dark regions, basically is your various genes are present. So like here, gene 1, gene 2 is present. Yeah. So, what is your deoxy? Yeah, DNA. Let's talk about DNA now. Let me just check if. Oh no, why I have to open like that? I want it to be in other way. Why cannot I open like other way around? And show. What? Sorry. So I have to always open it like that. Um, um, Yeah, so DNA is your nucleic acid that contains your genetic instructions that involves in various organisms and some viruses also they are present. So let's see the structure of DNA here. So this is a two long strand DNA makes the shape of a double helix. So like this, 5 prime to 3 prime end, 3 prime to 5 prime end. Two strands run in its opposite directions. Therefore, it is known as also called as anti-parallel. Just remember these underline lines. These are quite important from your examination point of view or objective type if somebody is preparing, because mostly questions are being asked from there. Uh, then chemically, DNA consists of two long polymers of simple units called nucleotides. So like this is your nucleotides. Backbone is made up of a sugar and phosphate bonds like this. And you have between adenine and thymine double bond, and guanine and cytosine is a triple bond. You can see it very nicely here. So what is difference between, this is sometimes we always get confused, what is the difference between nucleoside and nucleotide? So nucleoside is something with the sugar plus base, and whereas nucleotide, in which sugar is base is already there, just phosphate has been added. So just remember that. So if sugar and base is there, so keep remember with the sugar S you have nucleoside, but if phosphate is added then it's become nucleotide. So like this, you have a nitrogenous base pair ATCG. On the other side, your pentose sugar has been attached, and then on the pentose sugar, you have a phosphate group. So it's become your nucleotide. So in the bases of your DNA, so they are adenine and guanine, and cytosine and thymine. So your adenine and guanine, they are known as called as purines. You already might know. And cytosine and thymine, they are six-membered rings. They are called as pyrimidines. So here you can see adenine and guanine like this fused together. One is a hexagon, then is a pentagon, all together in this way, and in this case they are single benzene rings. The difference between your thymine and uracil is present in RNA is mainly the methyl group. Methyl group is absent from its ring. So pairing is like this: A pairs to T and G pairs to C, and whereas A pairs to U in the RNA case. DNA double helix is stabilized by hydrogen bonds. Yeah. So between the base attached two strands, like in this case, adenine and thymine having this single, a double, sorry, and in the case of guanine, cytosine, there's a triple bond is happening. Let me add just water, please. Just wait. So, thank you. Um, so what is the main difference between DNA and RNA? That is, on your 2' end of your carbon, there is a hydroxyl group is absent. So in case of deoxyribose in DNA, this OH group will be not present. In the ribose uh, nucleic acid RNA, so here your OH is present. So like this. Cool. That's the main difference. So this is your how hydroxyl group OH is added here. OH is being added there. Then here is a CH2OH is being added there, and then this oxygen bond is being attached in this pentagon structure. Same hold for the RNA, but the main difference is that the second prime end. So you have your hydroxyl group present in the RNA, whereas it's absent in the DNA. And this is your structure of your DNA. That looks like this is your minor groove. This is your major groove. Yeah. So the DNA chain widened around is around 2 nanometers or 222 to 26 angstroms, and one nucleotide unit. So distance between these nucleotide units is around 0.33 nanometer long, and the smaller groove here is known as minor groove, and the upper one is known as your major groove. And these are your various base pairs. On the side chain is your sugar phosphate backbones. So these are the basics, students, that you should be aware of, because from these only the main questions are coming up. Uh, if you remember the diagrams, you can answer things very clearly and very strongly, and your basics will be really stronger. Then you can answer many things easily. So try to focus on these slides, what I'm teaching today, absorb as much as possible, and when the workshop is finished, just go through you will be sent directly on your telegram after this, these presentations. Just before sleeping, just go through them, just have a skim eyes and sleep, and next morning when you wake up, just watch them once more. So you will see that you will remember these slides for your long term memories, for your long term future. Because if you get these learning habits of between sleeping before learning before sleeping and waking up the same thing. So they will be somewhere infused in your subconscious mind. So that's how all the medical students, all the big great scientists, if they're having a big things to learn, so it's very hard to absorb everything in our conscious mind actually. On the long term, if you want to get past in one night day, you can go like that. But if you want to be a long run horse, who or your marathon runner, so you have to work in a very systematic manner to learn the things. So to continue, so we have done with the RNA structure now. Now let's continue with the R yeah DNA structure. Let's continue with the RNA structure. So in RNA we can see it consists of nucleotide units. So here we have C, A, G, G, C, G. So each nucleotide consists of nitrogenous base pair, ribose sugar, and phosphate group. Like of here thymine, we have uracil, and it's not like double helix structure always. Sometimes it's double helix, sometimes single nucleotide. So it could have yeah different um, yeah bases and different way of working also. We also have double-stranded DNA. It's present in various viruses actually. This double-stranded RNA could be found in viruses, and also is double-stranded RNAs could be is the RNA with the two complementary strands similar to DNA found in all cells. So it's the same way as the DNA is, but it's found in the viruses. So various forms of types of RNAs in various modifications. So first is the messenger RNA, ribosomal RNA, and transfer RNA. So messenger RNA is dealing with the codes for proteins that all organisms it's in presence. Then ribosomal RNA is needed for the translation for your from your mRNA to protein when it's happening, and also the transfer RNA will be required there too for the translation. When after the then after the transcription is done, so in transcriptional modifications, we have small nuclear RNA, yRNA, and tolymerase RNA. So snRNA, it helps in the splicing and other functions. It's present in the eukaryotes and archaebacteria, whereas yRNA is present, helps in the RNA processing, DNA replications. It's present in the animals, whereas telomerase RNA helps in the telomere synthesis. That's on the chromosomes, telomeres. It's present in most of the eukaryotes. Then comes your regulatory RNAs. They are antisense RNAs called as aRNA. They help in blocking that translation or managing the mRNA stabilization or mRNA degradation, and they are also present in the all organisms. So something about mRNA. So mRNA carries the information of a protein sequence to the ribosomes. So it's present in the nucleus. From the nucleus, it is coded. Then codes. No, it's not coded in the nucleus. It just moves away uh, from your cytoplasm or from your nucleus to the cytoplasm. And it is coded so that it have three nucleotides codons corresponding to the one amino acid. So here you can see this is your single double pair DNA with the bases pairs. It called uncoiled. Yeah. After the uncoiling, you have your coding strand on that side. Yeah. Other is the non-coding strand which will not code. And then from coding strand for the C, G will produce. For the A, U will produce. From the A, U, T, A, G, C, G, C, C, G, T, A, A, U. Like this, you have messenger RNA being produced. And during this process, as you know, there's introns and exons are also present. So introns are the non-coding sections which will be removed. Only the exons will be come play in the role in this case. So we have discussed your mRNA exported from your nucleus to cytoplasm. So ribosomes plays role here. So your mRNA bound to ribosome, and with the help of tRNA, your proteins are produced. All text, I know a lot of text is happening so far, but it's quite important. If you have subjective exam, you have to tell sometimes things to be subjective to your teacher, to your exam questions. So, but I will also have some animation techniques. So all these things will be explained in quite easy manner in there in the later part. Okay, then further, in the prokaryotic cells which do not have nucleus and cytoplasm compartments, mRNA can bind to ribosomes where it is being transcribed by the DNA. So it's something different happening in the prokaryotes. Uh, Let me just check if you have any questions. So guys, help each other in the video while I'm giving the lectures. Help each other, yeah. Yes, yes, you will get my PPTs and PDFs after the lecture. That's no problem for me. Uh, mhm. Okay, so we are done with the DNA structure, we are done with the mRNA part, and now comes the tRNA. So transfer RNA is a small RNA chain of about 80 nucleotides chains, yeah, that transfer your specific amino acids to your growing polypeptides. So and it happens during your translation. So here you can see each tRNA has this intermolecular base pairing is happening within it. So A, U, G, C, G, G, C, G, C. And also you have your ester bonds being attached here with the amino acids on top. So from here your different amino acid chains will be made. Okay. And on the bottom this tRNA will be attached within the ribosome to make mRNA. So if there is a mRNA codon called GCC, so here you will have CG G. Sorry, it's GCC, then it will be CGG in that case. So it has sites for amino acid attachments and an anticodon region for codon recognition. So this is your anticodon region on your tRNA, and this is a codon region for your mRNA. And then that site specific to binding specific sequence on the messenger RNA through the hydrogen bonding. Okay. Uh, so now yeah, so tRNA is done, everything is done. Now comes your third part of your RNA, that is ribosomal RNA. So in eukaryotes, they are known to be present of four different RS RNA, that is your 18S, 5.8S, 28S, and 5S. These are subunits of your ribosomal RNA, different isoforms. You can see as the sizes, as the size looks like actually. So they are synthesized in the nucleolus. Okay, in the cytoplasm, a ribosomal RNA and protein combines to form your nucleoproteins called ribosomes. So from them, in cytoplasm, from your ribosomal RNA and protein combines to form your ribosomes actually, and these ribosomes further binds to mRNA. Yeah, they binds further to mRNA and carries out your protein synthesis. Several ribosomes may be attached to single mRNA at any time. So ribosomal RNA is extremely abundant and makes up to 80% of your 10 milligram per ml of RNA found in typical eukaryotic cytoplasm. So they are quite abundant present, quite a lot in cytoplasm. So after done with the various RNA different parts, DNA parts, there's a small difference between discussed here now between DNA and RNA. So RNA is your containing ribose sugar, whereas DNA is a deoxyribose. The base is here is uracil, on the other side is thymine. On the second position, your ribose sugar is present, where on that side in DNA it's lax. So RNA is usually single stranded and DNA is usually double stranded. So that's the main difference. It is being asked many times. Even in my interviews, I have been through some interviews. So I've been asked straightforwardly, give me a difference between DNA and RNA. So sometimes we studied so much, we forget to know the basics of this thing. So just to keep in mind, so this is quite important and quite important part. to be remembered. So guys, we are at the protein now. I will check how can I make this. Yeah, I know. Today is a basic, that's why it's a basic class, basic of molecular biology. Um we should know about how, because everyone is at different level, everyone is not at higher level of yours maybe. So I'm taking everyone together, and slowly we try to merge a complex thing also to it, and clear things for you also. So don't worry, we will do it, we will do it. Yeah, so guys, it's 2010. I will take a 5 minutes break, and then we see us at 21:15. Yeah. So I see you at 21:15. In meantime, just yeah, take water break, toilet break or something. I see you soon then. Yes, Sakshi, you can have access to the video later also. Yeah, no worries. I come in 5 minutes. Mm, So, guys, welcome back. I hope you're refreshed. Let's continue with the protein. Um, so, some were saying that this is quite basic and you're getting bored. So, don't worry. It's challenging sometimes when you know something. It's like mm, you're thrown into nursery class where you are told you have to sit and learn A, B, C, D, and you will be saying, "Why should I learn? I know already." But sometimes we forget things, and to start having foundation, it's good to remember our past, how we learn all these basics. So that's the main reason for today. So, proteins. So they are known as the polypeptides, made of amino acids. They are arranged in the linear chains and folded into a globular form. In the protein section, I had worked a lot with the proteins actually. My main PhD was dealing with the proteomics. Sorry, proteomics, where I did mass spectrometry and also did a lot of computational biology. I also did some Western blotting. So I will teach you all these techniques, how these techniques comes together and solve your mystery question. How all the things that you're learning in molecular biology and biochemistry techniques, how could they be beneficial for you? Yeah, how could be how could they be? This is disturbing. I have to close it. How could they could solve your hypothesis that you're looking for? How could they could answer the so unsolved mystery of science, from your ribosomal RNA to mysterious post-translational modifications, or even the folding of proteins, how folding of proteins is being done, which is being found in many diseases, such as recently like Alzheimer, or in the various neurodegenerative diseases, where people doesn't know why people in the old age they tend to forget things, like in the black film, have you seen, with the Amitabh Bachchan is forgetting everything at the end of his life. And this is happening in the our West world quite common, and it's coming to India also now. What is the reason behind all these things? So it's all happening because of proteins modifications, because of change in the proteins at the post translational level. So to understand that, let's first understand what is protein is, how is it made up of. So protein is a sequence of amino acids in a protein. We define sequence of genes which is encoded in the genetic code. So there are around 20 standard amino acids. So here you can see, yeah, today I was actually, yeah, eating guava, and also had some some seeds, pumpkin seeds also. So it remind me of amino acids actually. Everything is made up of amino acids, and amino acids changed together makes proteins. So whatever you are eating at the moment is actually made up of amino acids and further into the proteins. So whatever you're eating, it's in the form of amino acids actually. So just think before eating today's dinner, so that you are eating amino acids, a various long chain of amino acids having disulfide bridges like this here between cysteines and cysteines. That was my PhD project, to find out the disulfide bridges between cysteines actually. So I will also share with you, this is recent work done within collaboration with the King's College London and University of Göttingen together. And our university is famous for various Nobel laureates also. So this work was quite up to the mark and quite challenging for us also. So, what are the basic players in molecular biology? That is DNA, RNA, and proteins. So, what they do is they do DNA. From DNA, it could go for replication by itself to create its own copies. Then it could go for transcription to form RNA. Then from RNA, it could go to make protein via translation. And this whole process of story of from DNA to protein is known as central dogma. Remember that. This is quite important. What is central dogma? This is story of from DNA to protein via transcription and translation is known as central dogma. Then DNA replication is the basis for biological inheritance. It is a fundamental process occurring in living organisms to copy the DNA. Two DNA identical DNA molecules can be produced with the help of double-stranded DNA molecules. So here I can show you. I hope you remember this also diagram from your Okazaki fragments and how your DNA replication is being done. So this is your double-stranded DNA. You first topoisomerase will uncoil your protein, make a parallel chain of a DNA. Then helicase will come. It will break down your proteins into your bases, DNA bases supposed to be separated like this. Then from here, from this side, to start a new chain, your DNA polymerase will be added to make new chain for your amino acids. Yeah, and then with the help of Okazaki then there will be some Okazaki fragments will be made where there is no addition of proteins or no addition of fragments are there. These are no Okazaki fragments. And then further, your DNA primase will make your different various base DNA base pairs. Yeah, and then like this, your whole things will be going on with your DNA replication. Now comes the transcription. Transcription is the process of creating an equivalent RNA copy of a sequence, as we discussed. So from DNA, you can make RNA. From RNA, you can also make DNA. That is known as your reverse transcription. So during transcription, a DNA sequence is read by RNA polymerase, which produces a complementary antiparallel RNA strands. So in transcription, RNA complement includes uracil instead of thymine. This we already know. So this is what we have discussed, how transcription process is being done. You have your RNA polymerase is being attached. They are uncoiled. So there's a promoter sequence like AA, CT, GT, AT, AT, ATA, and you have a start signal here. And then RNA polymerase go from left to right, and then you have your self-complementary sequence being produced, and hence at the end your DNA strand with the help of coding strand. Your complementary RNA is created in the 5 to 5 3 prime direction. And we have discussed already exons are the one where we can your your RNA synthesis and processing is done, whereas intron they are the non-coding zones, exons are the coding zones. Your introns will be removed from them, and then hence at the end you have your exons only, and RNA synthesis and processing could be done in that case. We discuss also reverse transcriptase, reverse transcription in which like your HIV RNA is there, which is being reverse transcripted with the help of reverse transcript enzyme. It forms into the HIV DNA. It goes back into its back form. So like from DNA, RNA produced, from RNA, DNA could be produced with the help of enzyme reverse transcriptase. So this is quite general, but this is special case like this is happening. And also if RNA is producing it by themselves by replication. Now comes the last stage, that is your translation, which is the first stage of a protein biosynthesis. It's happening in the ribosomes located. So here you can see very nice diagram where valine, alanine, and glycine. These three is on the messenger RNAs within the ribosomes. They are attached here. You can see. Yeah. So what will happen? A tRNA will come here. So for like say tRNA of valine was here. Yeah. So valine came and anticodon got attached there, and then the transcriptase has valine protein up amino acids. So it got attached to this protein chain of amino acids. Then comes the alanine. Alanine has this our amino acid attached to its transcript tRNA, and it's got attached to it, and then it will remove afterwards and then further go. And then next with glycine, and this way your whole protein amino acid chain will be produced with the help of your ribosomal RNA. So this is quite important to remember your various genetic codes UCAG. You can see that here, UCAG, and also you left hand side you write UCAG. These are your first base pair. This is your second base pair, and then on the right side you write again UCAG, UCAG, UCAG. Then on the third, so yeah, like this you write all of them. Then you write like this U. Then on the top you take U from here, U, then again U, then you take C from here. So this is these together known as your phenylalanines. Okay, these are known as phenylalanines. Then further UUA like this, and then UUG leucine. These are called leucine. By this way, remember them. It will be really good for you in future. If you could especially remember the stop codons like UAA, UAG, and UGA. So these three are your stop codons. Remember these for sure. And if you can learn more, that's always good for you, like serine, arginine, glycine, aspartic acid, alanine, valine, methionine. And methionine is your starting codon, AUG. From AUG, your mainly your translation will start and it will end at these three stop codons. So remember this genetic code. So what is genome? Genome is so we have discussed so far about what is a DNA, RNA, various types of RNA. Then we went for protein side, how difference between DNA and RNA. Then we discussed about transcription, then translation, and also a little bit about replication. Um, and then recently we discussed about translation at the end and genetic codes. Now we are coming into the genome part, like how a genome is your entity is being generated. So genome is entirely organism hereditary information. Uh, so it is encoded either in DNA or for many types of viruses in RNA. Genome includes both genes and non-coding sequence of the DNA. Yeah. So further, comparative genome size, you can see from bacteria, influenza, and Escherichia coli, the size base pairs from the Homo sapiens, mouse, Drosophila, how it's being drastically different. So in the fruit fly, it's 137 million base pairs. So it's a very good model for genetics people who are studying genetics. If they are looking for genetics questions with various Punnett square and all those methods, so this fruit fly they are using it in their research. In the plant section, they used as a model is Rhabdopsis thaliana, has around 100 million of size base pairs, and gene numbers are around 25,000. So average gene density is one gene per 4,000 bases, and chromosome number is 10. In the bacteria, it's 1. So this is just tell you to see the difference between different species. So these all things are the um, just these are the various uh, yeah, various types of genome size in it. So next is why genome analysis, the prediction of genes in uncharacterized genomic sequences. So we can characterize how it has to be happen. We can make new varieties at very faster rates like BT cotton and BT brinjal, because once we know the genome of one species, like for human being it took a lot of years to do so, but if once we know of like any cotton or any species, corn or any vegetable or any fruits, we can try to manipulate the genes very easily and try to bring new species out of it. So with the help of transgenic plants or transgenic animals, like from Agrobacterium tumefaciens, in which we do genetic engineering to do so. With the help of cloning also, we can create. So, basics are done. I will stop it here now to take you to the journey of a bit excited part. Uh, of not gene therapy, not this one. So this was quite a lot of text and lot of information. So I want to take you to a video. So what are proteins? So these videos will explain my topic that I have taught you now uh, in a more animated manner and in a more versatile manner actually, so that you can understand the concept what we have learned in that thing and in this manner. So I will start.
Proteins carry out most of the body's functions, and they make up the bulk of its structure, from hair and fingernails to bone and muscle. In fact, nearly everything in our bodies is made from or by proteins. The instructions for building proteins are found in segments of DNA called genes. Proteins are made of long chains of building blocks called amino acids. There are 20 amino acids in all. Put together in different combinations, amino acids form proteins with a broad variety of shapes. This diversity of form gives proteins their many functions. The proteins a cell makes determine what it can do. Our cells are constantly at work, producing the thousands of different proteins that keep our bodies functioning.
So something about protein.
Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. During translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. These proteins perform the tasks that make living things function. The flow of genetic information from DNA to mRNA to protein is central to life. Inside the nucleus, RNA polymerase attaches to a gene. It separates the two complementary DNA strands, exposing the gene's unique sequence of bases. When the end of the gene is reached, transcription is complete. The new strand of mRNA passes out of the nucleus and into the cytoplasm, where translation takes place. The instructions in an mRNA strand are read in groups of 3 bases called codons. Each codon must be translated, one after another, in order to build a functioning new protein. The key structures in that process are the ribosome and transfer RNA, tRNA. Each tRNA molecule has its own anticodon and a matching amino acid, the building blocks of proteins. To begin translation, a tRNA molecule coded for the amino acid methionine docks with a ribosome. The ribosome attaches to the end of the mRNA strand. It slides along until it finds a start codon, AUG. Through complementary base pairing, the start codon pairs with the mRNA molecule, signaling the beginning of the protein instructions. The methionine molecule is now the first amino acid in the new protein. The ribosome shifts to read the next codon. Each codon is recognized by one of 20 different tRNA molecules. Each type of tRNA carries a different amino acid. The ribosome continues moving along the mRNA strand, one codon at a time, matching it to a complementary tRNA. It adds each amino acid to the growing protein in just the right order. The sequence of amino acids determines the final shape of the protein and the function that it will carry out. Give translation a try. When the ribosome reaches a stop codon, it detaches from the mRNA strand. The chain of amino acids folds into its final form and the new protein is complete. This protein, aminoacyl tRNA synthetase, will now attach the right amino acid to the proper tRNA molecule, charging the tRNA so that it can participate in protein synthesis. Each type of amino acid has its own enzyme. The incredible processes of transcription and translation enable cells to carry out all of the functions necessary for life.
So, class, was it interesting? Um, so the reason behind for showing you um, the videos and the presentation before, so to make interaction together, so you can understand what we are studying in such books, how things are, and how practically they are done when we are about to study them. So that's the main idea. So Let's continue with the tools now. So today I will discuss mainly the all the tools. Uh, so tools used in molecular biology. Basically, whatever the techniques that we're gonna learn in our upcoming time. So I just try to make it a simple, a little bit gist about them. So we will not go through in details about them, but a little gist so that we can have a view like how things are working in it. So let's start with first the gel electrophoresis. So this is the basic principle is that DNA and RNA or proteins can be separated by means of electric field. So one in this we have in the gel, one is the negative electrode, one is the positive electrode, and we had our DNA in it, which got separated because of the charge is to size ratio. If somebody asks you that how DNA in a gel electrophoresis, on what basis it is being separated, so mostly we say that it is size, but we have to say that its charge is to size ratio. And proteins could also be separated by using SDS page, whereas in the DNA it's being separated by DNA gel electrophoresis. Uh, and also in protein we can differentiate on the basis of the electric charge by using 2D electrophoresis also. So there's a 2D, so this is part of proteomics. So the omics is something, it's recently being very famous. Omics like transcriptomics, proteomics, genomics, uh, yeah, all these omics parts, they are actually because of the study of this these uh, yeah, omic studies is famous for. So the transcriptomics also coming up there. There's a post translational modifications also coming up there. So all these things makes a lot of sense actually. Now PCR, which is quite hot in our everyday day life, it makes life quite easy. It did a lot help in my PhD also, and later part also after my some work I did in the labs. So polymerase chain reactions, they even at the moment the COVID-19 tests, they are based on your PCR simple test actually. So they have RT-PCR machines, and they just check, they take from your sputum, from your nose, the DNA from your throat and from the nose, and then they just get the complementary DNA out of it, and then just put in the machine, and it just run in the machine and just check for is it positive or negative then. So it's all based on because it's increase the copy number of your DNA, because we have very less amount coming up at the first. So to increase the copy, mainly PCR is there. So this is a little bit just about how your PCR is working actually. So this is your template DNA, yeah, which producing your first cycle, and you have different temperature. Initially, it is around, so far I remember, 30 to 40 degrees Celsius. Then it goes to 72 degrees Celsius. Then the temperature is again decreased. So this is DAE, degeneration, annealing, and elongation. So three things are happening in this. So this is your first cycle in which you have copies of 4 copies, then 8 copies, 16 copies, 32 copies, and then after 35 cycles you have around 60 billion copies. So this is how exponentially amplified from your just small template DNA you can produce such a huge amount there. In this case, it is told that why primer is important. So we will not, I will not today go much in details about techniques. This is just about various techniques that are being used um, in the molecular biology techniques. So this is various applications that you can use for. You can go through them. Then this is to do a micro molecular plotting of probing. Yeah, how you can blot big molecules of proteins. So this is or genomic DNAs. How you can do a big large molecules. So this is the technique has been shown here. It's called as also as a southern blotting. So you can see we have a genomic DNA, and we restrict it with the restriction endonucleases. This is a kind of a Caesar. Yeah, it cuts your DNA into different parts, and then After the agarose gel, you have your high molecular weight and low molecular weight. Yeah, so they have one is positive side and negative side. So it will transfer. Then after the transferation of the on the gel, we put it on the blotting paper on the, and with the help of nitrocellulose membrane, we transfer your protein onto it. Then after it is being transferred on the nitrocellulose membrane, we have our radioactive probe, phosphate 32, on this. On the other side, then whatever the genomic DNA that we are looking for, this probe will attach to that part, and this will be exposed to X-ray film, and we can extract this part from our gel then later on, and we can do our things that we are looking for. It's just tell about the same southern blotting. So we are talking about now various southern blotting is done for DNA, northern blotting is done for RNA, and whereas western blotting is done for your protein sections. So I will be dealing, so I have expertise in the western blotting. I did this a lot in my PhD. So I will be sharing what are in and bits about it, various protocols of also, and we also go through very nice video about it. Yeah. So in the Western blotting, uh, yeah. So so in the Western blotting, it's mainly done for the proteins with the help of SDS page, that is sodium dodecyl sulfate polyacrylamide gel electrophoresis. So here also, after your gel has been run with the help of on various wells of the protein, it is transferred to the nitrocellulose membrane, and then with the help of antibodies, we visualize in the chemiluminescence or the autoradiography. So we will go. So no panic. This is just a gist about giving you that about various techniques. We will deal with them. Then various molecular markers like RFLP, AFLP, SNPs, microsatellites, all these markers, they are also quite helpful to check the polymorphism in DNA sequences. So they are quite happening like base pair deletion, substitution, addition, or pattern is happening. So this is also quite important technique in your yeah, from examination point of view, also from others point of view, your objective type or subjective. So further, bache, these are the various definitions. They come quite commonly in your exams. RFLP, restriction fragment length polymorphism, AFLP, RAPD, VNTR, microsatellites, SNP, STRs, SFPs, rad markers. So all these techniques are being used there also. So remember uh, their full forms. They're quite important. So some correct how to characterize a suitable molecular marker. So these are the various things have been shown there. Um, yeah, just go through the applications. So this is something about what is RFLP is doing. Its name says it's restriction fragment length polymorphism. So from the DNA, a sample has been restricted with the help of has been cutted with the help of restriction enzyme, which can recognize and cut a specific DNA part of that sequence, and then this can be detected, and it varies from various individuals to individuals. So advantages are it could measure variations of level of DNA sequences not in the protein sequence. Disadvantages is relatively large amount of DNA is required in that case. Whereas amplified fragment length polymorphism, in which we amplify the restricted fragments, the fragments that were restricted, we amplify them and then analyze them to comparison between the various closely species. Thus go through all these techniques. So mainly here, important part is SNPs actually. The single nucleotide polymorphism, which is a DNA sequence which is occurring whenever the single nucleotide of the genome differ from the members. So like CG is the normal case, but somehow, sorry, ATA is the normal case, but somehow it's changed to the CG. Your purines are transformed into the pyrimidines. So in that case, you can see a quite drastic change at the functioning level. So this is we use in various biomedical research, crop, livestock breeding programs. So within the if we want let's say, salt tolerance plant. So where the place the plants doesn't need a lot of salt. Yeah. So what you can do is just transform these base pairs. Yeah, just transform these base pairs. So you can change. You can have that effect that you're looking for. So this is all done with the various techniques to know. So for that, again, if you want to do this, you should know the genome of that species, or whole genome, and where you can do this mismatch, where you can do this deletion, and then leads to your desired effects in it. Then comes the STRs, short tandem repeats, STRs, in which you have a some like 4 units like this, 2 to 16 base pairs, in this case 4 base pairs here, CATG, they are being repeated after sometimes, typically in the non-coding intron regions. So it could be used if they are being repeated, used in forensic cases or genetic fingerprinting. This is quite interesting. We will do this later part, genetic fingerprinting also. Yeah. And further, principle of DNA isolation. So we are done with the various RFLP, AFLP molecular markers techniques, some northern protein techniques, DNA techniques, PCR, and then gel electrophoresis. So today I will just tell you now to start with, have a little bit of to start with first technique, that is your DNA isolation. So what is the principle behind this? What is the how DNA can be isolated from the nucleated cell and the giant anionic solution? So source of DNA could be blood, buccal cells, or mouth cells, culture cells, bacteria, biopsies. So we can use all these various specimens for your DNA. So to do so, it's a quite common molecular analysis. So basically, it's divided into three different parts. That is cell disruption, isolation of DNA, and precipitating of DNA. Okay. In the first case, we disrupt the DNA. Uh, we remove the sorry cell. So this is commonly done by the sonication. That is with the help of a large sound, a high sound frequency, or by grinding it the sample and removing the membrane lipids by adding a detergent, and lipids are removed with the help of detergent. So this is your kind of cell lysis is happening here. Then isolation of DNA, removing the proteins by adding a protease. So protease will remove your proteins out of it, so you will have mainly DNA left in it. Then precipitating the DNA using an ice called ethanol or isopropanol. So we precipitate this at the bottom. Since DNA is insoluble in these alcohols, it will aggregate together and gives a pellet. Will have a pellet at the bottom, and this steps also removes alcohol soluble salt from it. So your all other salts will be also removed with alcohol soluble. So basic rules, what we have to do for the in case of blood, if we're taking blood as a sample, first we have to lyse the red blood cells with a gentle detergent called as Tritone X100. This is quite uh, available in the laboratory. We just add some amount to it so you can lyse your red blood cells. Then you wash your cells uh, like hemoglobins, other which could inhibit our restriction enzymes and Taq polymerase. So we have to also deal with that part. Then we bring the enzymatic process to be slow. So we bring them into the ice, and while doing so, please wear the gloves. Yeah, and autoclave all the solution is stored in the fridge. Whatever you're working with your things, please autoclave them. Yeah, and keep all pilots and supernatants until you have the DNA you want. Yeah, just keep it everything separated. So getting to the DNA. So how you do it? First, cell will be lysed in the presence of NaCl, so that DNA is stabilized. So this is quite important. This figure is quite important from your principal point of view. So what is doing? This is mostly being asked in your oral examinations or wherever you go. So which part is doing what? So this is quite good summary here. Which part is doing what in DNA isolation? That is uh, NaCl is cutting. NaCl is making your DNA stabilized, and remains double helix in that part. Yeah, your salt. Then EDTA is added as a chelating agent, which chelates magnesium plus plus magnesium ions and is a cofactor of DNAs which chews up the DNA rapidly. Okay. Then anionic detergent SDS, which disrupts the lipid layers. All the lipids are disrupted and help to dissolve your membranes and bind positive charge of chromosomal proteins to release DNA into the solution. Okay. Then further include a protease to digest the proteins. Protease is added to digest your proteins, and we can incubate the solution at the elevated temperature, that is 56 degrees Celsius, to inhibit the degradation of DNases. So that we do not have DNases to be inhibited in that time for 24 to 24 hours. Then we can remove this protein by the using volume of phenol into chloroform 24:1 ratio, and then precipitate at 10 degrees Celsius for 10 minutes at 10,000 rpm. Then after precipitating, this is quite, I mean, this is very straightforward. You will have a protocol in your hands, you will have your DNA, and you have to do the things as per suggested to you. So this the main reason to show you this table is to understand, so which when you are adding the things, when you are adding the EDT or protease, or why you are increasing the temperature, why you are adding the SDS, why you are adding NaCl. So all these things will be, you get to know that what is the reason why we're doing these experiments. So always remember, when you're doing experiments, just ask yourself why, why, why we are adding that, why we are not adding that, why this was possible. If I added this, what will happen? So it's like if you're making, I will take another analogy. Instead of in making your Maggie, instead of adding Maggie masala, you add lot of sugar. How your Maggie will taste? It will taste horrible to me. It will, I don't know if somebody likes that. So it's just same way, instead of adding EDTA, you add some other thing, so your whole process will be changed completely. So it's always remember to the recipe, how the recipe is working, and to get the data that you're looking for to be right way. So you can precipitate then the DNA with these methods further by adding ice cold 95% ethanol and leave it at minus 20 degrees Celsius overnight. Then centrifuge it at this rate for 10,000 rpm for 10 minutes at 4 degrees Celsius. Then you can remove the excess salt in 70% ethanol hydroxide and air dry, resuspend in the sterile distilled water, and then you can store this at 4 degree or at minus 20 for long term. Sorry, my voice got disturbed. And then further you can quantify the DNA, how much you have, how much DNA is being produced with the help of spectrophotometer, and this using this formula in it. Then you can also check the purity, that how much you have protein contamination or chloroform presentation in this. That's it. That's how the DNA extraction is being done. The summary of it. So then thank you so much for today, for your patience, for your being tolerating me. For almost still 10 minutes are left. Uh, uh, Still 10 minutes left. So I want, so whatever the DNA isolation of DNA we have done, I want this to be done now. So you can stay just 10 minutes. You have to stay 10 minutes more because our class was 10 minutes late also today. So we continue this class for the, so guys, this is I'm using this platform, Learn Genetics, in my most of my teaching, whether I'm teaching in the university or I'm teaching at the personal level, because they are taught in very even I will share with you the link. I will share this link with you on this live stream, so that go through all these things. It's very useful. All the things that you're learning here, they have explained in very easy and very simple manner. That's the beauty of sciences. That's, if you can understand simple, you can teach simple. You can apply that in your daily life. Yeah, so I will take you to the next part of our workshop today, that is DNA extraction. Because I cannot take you to the lab now, so this is the best way they have shown us to teach you how DNA extraction or biotechnical you can DNA extract in this virtual lab. So I will unmute myself now. Oh, sorry, I will mute myself because I lab now. So I think there is not so much voice is required because I have to unmute my computer voice, otherwise it will echo for you. If I play sound from my laptop, so things are different. So far, did you understand what we have done? So we had took our DNA from the swabs and then we added lysis solution to break down upon our DNA cell wall membrane. Then we are breaking it up the nucleus part. Then from the nucleus we have this histone part being there. So this could be broken again with the help of protein kinase K, which is a detergent here. So this will unwrap our coiled DNA from the histones. So this was done while adding to the water bath. So we have it a good temperature, warm water in it. Now we are adding concentrated salt solution to it. So what is this doing? It's after coming from the warm water for long enough. So cells has to be free now. So we add some from the tube. So add some concentrated solution to your tube to do so. So you can see uh, your cell debris has been removed, the protein debris has been separated, and your DNA is separated out of it because of adding your concentrated salt solution. Now we will microcentrifuge it. So always add on the other side also to keep a balance of each other. This you might know. So click the lid and let's start. So what we see here is proteins and debris at the bottom, yeah, and DNA liquids at the top. So it is being differentiated because of this centrifugation. Now we will take the supernatant from the pipette and add it to our fresh tube which we have only mainly DNA. Now it's time to concentrate isolated concentrated DNA by adding isopropyl alcohol as we discussed in the presentation. So we add some alcohol to it, to the tube. There it goes. So with the help of it will precipitate. All DNA will clumps together and looks like this. Clumps of DNA with the naked eye. We can see the DNA to be present like that. So we put this again in the microcentrifuge and close the lid. So at the bottom is your DNA. So that's it for today class. It was too fast. So for today you have to just make a summary assignment, just 2-page summary, whatever we have done today. So maybe I can send you everything attaching files of today. So you can attach this objective first of all. And workshop day 1, these two presentations. And I will save the video now after this session, and we can go for further things. So any doubts, any problems you have today, so we can start tomorrow with a fresh start tomorrow. Yeah. So I will yeah, it's just because of lack of time. I just I have to go like this. Uh, so today there was yeah, initially we had some buffering problem, but later on it was fine. So So it's everything is there in your thingy in your Telegram now. Um, so I will write on the Telegram about write it down on the Telegram about your assignment thing, how you have to give your projects or assignments. So yeah, it will be just two-page summary about molecular cell biology, basics of molecular biology and cell. So just try to merge everything together you can. Try your best. Okay. So that's the end of the class. Yes, bache, it will be nice if you can draw one diagram of model of a factory too in that. So we have this testing, to check the purification by spectrophotometer and to check the polarity. You can check it as we discussed in the presentation. So I will tell about assignment in the Telegram, like what you can make. So basically, I will again tell. Yeah, yeah. So tomorrow we will be. Today was the basic session. So just to tell what is the basic idea about DNA and RNA. So we will go with each techniques tomorrow, one by one, and go in detail about it. So don't worry, don't worry. About your assignment, I will again repeat that you have to make an assignment of the summary of your 10 days workshop. Each day you have to write two-page summary, and in total it will be 20-page summary, and do it in your desktop, not handwritten, and send it on my email ID. That is, I will share it here, simranrihals99@gmail.com. So this will be done after 3rd of September. So 10 days will be done after 3rd. So from the next day onwards, from the 10th of from the 3rd September, you can start submitting it. So today is 25th is the first day. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Third will be done, and from fourth onwards, so you will be given 2 to 3 days. I think 2 days will be enough to send us your. So keep every day write 2 pages of summary in your WordPad and try to save it and just send me. So do whatever you learn, whatever you have done on that day, it has to be there. Um, Sorry, I will be happy to receive handwritten. It's easy, I know, but I will be happy to receive a proper format, like you're submitting your assignments, uh, not just any like just writing in your hands. By writing your hand is like sixth standard uh, homework we are sending. So no, please. Okay. So there are a lot of unnecessary messages on. Yeah. That's it. That's it for today. And at the end of the lectures, we also have to give feedbacks. Not today, by the end of our days. So give your honest feedbacks, whatever you feel like, what needs to be improved, what things could be done better for you. Please tell us about that also. Yeah. It's up to you. Format is not necessary, but it's up to you how you want to speak about it. So, okay, guys, I have to leave now. Thank you so much for your time. See you tomorrow at same time, with same energy and same power of energy. Yeah, from all of you. Thank you so much. Sleep well. Take care. Stay safe. Stay blessed. Thank you so much. Bye. Sorry. What about? Bache, there is a projects. Yeah, yeah, projects, assignments is same actually. It's same. Projects and assignments are same. So don't worry about it. Thank you so much for your good comments. Thank you so much. Yes, yes, assignment is necessary. Assignment is necessary. Without assignment, you will not get any certificates. Mail ID is above. Please check in the chat. I will also share on the Telegram my email ID. And guys, you are around 350 students, so answering, don't if it is very important, only then write me personally. Otherwise, write me on email. I can respond to you by email very well, rather than you writing me on Telegram with short messages. It sometimes feel hard to have. If it is very important, write me your questions. I will get back to you, and we will try to solve your things, okay? um, and I'm here for you actually. Whatever the problems that you're facing, anything in the academia field, in your questions, I'm always there for you. Just feel free, ask to me, and we will go for that. Yeah, please, summary not to be handwritten. I will be happy to be typed one in the WordPad, because handwritten is something like sixth class standard homework is being done. So I will be not happy in that. So yeah, yeah. Okay, guys, I have to go for sleep because I have to wake up early tomorrow morning. Uh, Thanks. Okay, it was nice chatting with you, nice talking to you. I will see you tomorrow. Good night, take care.